All right, hello. So this is our lecture looking at temporal ecology. And we're going to mostly talk about short timescales, but this is coming from the Morin Community Ecology book, specifically chapter 9. So you can use that to help you out to flesh out some of these details. So temporal ecology, ecology due to, taken over a time scale. Is a really active field, and there's lots of things that we can discuss when we're thinking about how ecological processes happen through time. Now, what we got to remember is that it's multiscalar. So when we're thinking about, on the short-term side scale, we're thinking about things that happen, over the course of a day, things that happen on a season. What you can also have is this thing called community assembly, where you have species colonizing an area, and it's maybe a new habitat, or maybe it's like a lake that unfreezes and in the early part of the spring. And so we'll be talking about community assembly too. We're not going to talk so much about succession, so long-term, potentially hundreds to thousands of years, as you go from primary succession here, and how the community basically is built through time. Community assembly rules can somewhat talk about that, but we're going to spend more time thinking about the shorter term stuff. So when we're thinking about ecology, we got to think about, okay, do historical processes matter or do they not? So one way we can think about that is think about a new habitat. Does colonization order matter? Now, a lot of our older, no, I shouldn't say older, but the models that we've talked about in class so far basically are what we would call history-free models. Okay, so this is the Tillman R-star model, right? And what we've got here is two species, the yellow species and here that blue species. Now, given this model, if yellow colonizes first, right? There should be, a whole lot of resources, right? In an empty habitat, there should be a whole lot of resources out here. Now, those resources then will get drawn down to this R-star point for the yellow R-star point, right? So resources would be right here. But then that means that blue can come in and basically take over, right? So the blue species is a better competitor, and it can colonize if resources are right at this level, right? Blue can colonize. So in some senses, it doesn't really matter when blue colonizes. Blue is always going to be able to win. So the Tillman's R-star model in some senses really doesn't look at history of colonization order. It just thinks about species being there and then they reproduce. Or not, right? But realistically, the R-star model is itself a historical process, because think about what happens if the blue species were, let's say resources are again out here, and if the blue species then were to colonize, it would bring down the resources all the way down here, and then since the resources are at this level now, yellow would never be able to colonize. So Tillman's R-star model is in itself a historically based process, but we just don't kind of usually think about these things that we call priority effects. So what a priority effect is that species present early in the community development influence later arriving species. Okay. Now, classically, we have more information on short time scale processes just because it's easier to study, right? We don't really necessarily know how priority effects work when we're thinking on the 100 to 1,000 year time scale, right? Because people haven't been studying ecology that long. So we know a lot more info about short time scale kind of priority effects. So we can kind of classify priority effects into three different things here: facilitation, inhibition, and just no effect, right? So facilitation is one species that will help other species. Inhibition is, one species preventing other species from colonizing, or no effect, right? Now, when you put all these things together, what you get is what's called phenology, and that is the seasonal patterns of abundance or activity. So when we're thinking about maybe a year or the course of the seasons, we have the phenology of plants or frogs that come out in a pond, or any type of species that we want to talk about. When we put all these priority effects, we can create phenology, which is that pattern of species appearing in an environment or being active in an environment. The thing is, what we got to remember is, we see this clear pattern here of the phenology of, species coming out from March up into October. But so oftentimes they're just completely unrelated to priority effects, and we'll kind of talk about that situation here in a little bit. So what causes these phenological patterns, right? Why do we see certain species coming out and disappearing at specific times every year, or within the creation of a new habitat? And that's due to these things: temporal resource partitioning, facilitation, physiological constraints, resource availability, randomness, right? There's a randomness component to this. And then basically all of the above, right? In any sort of situation, generally what we see is that, one of these things is not just working alone. You have to get all of these things put together to create some sort of phenological pattern. So the first thing is, one of the reasons we see the phenology is temporal resource partitioning. Okay, so species can coexist using same limiting resources at different times. Now this might seem familiar to you. Think about that temporal character displacement that we see, that you can get. We talked about that during the ecosystem function unit, where we were looking at how this one resource, that flower, can support 4 different individuals here, 4 different species, due to the times in which they use that resource, the flower, the nectar in that flower. So what we're kind of thinking about for temporal resource partitioning is like that on a larger, pretty much the same thing, but on a larger time scale. Okay, so think about if the phenological pattern is essentially here. What we've got is, this red species is the first species to come out. So let's put a time axis here, and the red species is the first to come out. And then the yellow species comes out later, and purple species is last. Now, in here, what we have is basically them all coming out at pretty much the same time. So what we would expect for resources is, this is kind of crappy for resources. Like the purple species is kind of screwed, right? The purple species only ever sees the resources when they're right here. So, this isn't, in some senses, if you were, trying to design a system, this isn't the way you might want to do it. And through natural selection, some organisms have figured out that, okay, if we spread out, right, or if certain species are selected that have spread out phenological, like distinct periods, we can use this black line resource, whatever the resource this is. Let's say these are different plant species and its nitrogen levels, right? This black line then can stay up, and like during this time from here to here, there's time for that resource to rebound, to go back up. And then it gets driven down and goes back up, as we see the influx and then the senescence of species, repeatedly throughout the season here. The thing is, for this to happen, you really need a quick rebound resource. So if that black line resource, were to be really slow coming back, and it would just kind of stay back down at real low and then come back once everything goes away, this temporal resource partitioning wouldn't work, right? You need the resource to be quick rebounding. The thing is, temporal resource partitioning is really tricky to prove. Basically, the dispersion that you would see, so having all of these species spread out here, needs to be larger than would be expected by chance, and that's actually really hard to prove, and it's really hard to see whether that is working or not. What we do see is this little seasonal succession of phytoplankton populations might have to do with some of that. Certainly with the resource availability of diatoms, what's really important to them is silica. And that's basically just glass, and that's what they use to do their cell walls. And we can see that they are able to come out really early and get in really high numbers, and then they go away due to that silica not being able to be there. And what we see is that there is in this, we'll talk about this a little bit later, but there's a variety of different processes that allow these different species groups to increase and decrease throughout the season, but a lot of it has to do with resource availability, that being silica, and then we'll talk a little bit about light here later on. All right, the second thing I want to talk about is facilitation, right? Facilitation can cause phenological patterns, right? Now, this isn't that much really different than so I put here the slide from the beneficial interactions unit, right? So, one species aiding the establishment of new arrivals. How is this really different than mutualism or that true facilitation, right? Where one species enhances growth, survival, or reproduction of a second species, right? And essentially, when we're thinking about when we were thinking about facilitation in this scenario, right, we have foundation species of this sphagnum moss here, or the beavers going on in this dam that are ecosystem engineers. We're kind of thinking of that as just a continuous thing that's happening, right? The sphagnum moss is always facilitating the growth of other species in that bog. Whereas when we're talking about facilitation, what we're more in the temporal ecology sense, we're talking about how an early arriving species can affect later arrivals. So just a really easy example here is primary producers are needed to basically allow for herbivores, right? And an herbivore isn't going to be able to come into a new habitat. Think of like a new volcanic island that erupts from the middle of the Pacific Ocean, right? What's the first thing that's going to be able to survive there? It's going to have to be a primary producer, right? Because there's no food available for that. Now there's always exceptions, like a bird, a seagull that eats things from the ocean could just use it as a nesting thing. So that could be an example of a predator coming in first. But what we see is, if organisms where the food web is based on the island or on wherever this completely based within the ecosystem we're talking about, you would need primary producers first, which would facilitate the colonization of herbivores, and those herbivores then would facilitate the colonization of predators. What's interesting is that a lot of temporary ecosystem things and a lot of the work that I've done with my playa wetlands has shown that this isn't the case, right? You can have predators coming in first because they can basically get ready for the habitat, or once they find a habitat, they can sit there and wait because of the way the organisms grow so quickly. Oftentimes the predators will come in first and just wait for the prey to hatch within the next 2 or 3 days, and then they do come, and then they're available, and those predators are like, just sitting there waiting for the prey to come out. Temporary ecosystems, and we'll talk about them later in this unit, kind of break a lot of the what we would consider rules in when we're thinking about assembly or priority effects, because their time scales are so short and not what we're used to thinking about. Another reason we see phenology then has to do with physiological constraints, right? So these are, this is a little graph looking at basically when is the breeding period of frogs. And usually what we see is frogs like basically come out in the spring and are ready to mate right away. And why do they come out in specific times? A lot of it just has to do with their physiology, right? So I forget where this was. Obviously not somewhere up north, because we're having frogs coming out in late January. But so this is this is in North America, a little further south. But there's several species: a wood frog, spring peeper, little grass frog, come out very, very early in the season. Their physiology is such that they can deal with the relatively cool temperatures and can really easily live in February, March, and be active in January, February, March. Whereas other frogs, just like the leopard frogs, which are a little more northern species, just don't have that ability to come out until later in the season. It has to do with their individual physiological needs. So that can create some really interesting patterns, just due to when species are able to come out. The thing about this phenology is that it's really an interesting study, or studying phenology is really interesting now, just because we live in such a changing world. Okay, so when you look at say just two examples of a predator, a pollinating bee. Let's say this bumblebee, okay, and this daisy. These two species are codependent on each other, right? The bumblebee is codependent on the flower for nectar and pollen. The flower is dependent on the bumblebee for pollination, right? Without one, the other one basically dies. So what we see is flowers, most plants, really their appearance is determined by light levels, okay? So the thing is about light levels is it's a really good cue to use about when to come out or when to not. So a daisy will only come out, let's say, April 1st. Okay, why? Because the flowers essentially, through evolution at least, know that April 1st has enough sunlight, right? The day length is long enough. And the thing is, that's unchanging, right? The day length on April 1st, 2020 is the same as the day length of April 1st, 1900, right? So, over extremely vast time frames, that is unchanging. The amount of light is unchanging. The thing is, we live in a global warming scenario where that the temperature cues are getting different, are changing, right? And what we see is a lot of insects, a lot of pollinating insects, their appearance is determined by temperature. Okay, so they, are basically kind of calibrated to come out and start being active, let's say April 3rd, when an average temperature gets to be, whatever degrees, 45, 80, or 45 to, 50 degrees or something, right? And then the bumblebees can start coming out. But what's going to be happening then is wrong button, sorry. as the temperatures, as basically spring starts coming earlier, you're going to get these bumblebees coming out earlier. Now, for all these bumblebees that come out at this time, or up to about here, where are the daisies at? They're going to be gone, right? So all these early coming out bumblebees are going to die off due to the resource not being there, right? Because the daisies are not going to come out earlier because they're based on light. So we have these pollinator and mismatch, flower pollinator mismatches that can really have a big impact on populations here in the future. And the question is, will species be able to evolve fast enough to can the bumblebee evolve to basically come out later, or at the same time as it used to? That's a question that's still up in the air. So really what we see is this phenology is really an integrated behavior, okay? And it's integrated behavior through long-term evolutionary pressures. So diatoms are, due to their physiology, are able to come out really early. So let's say this is a Wisconsin lake, okay? And so we know ice, goes away completely somewhere, in March probably, maybe, let's say this is a northern Wisconsin lake. And diatoms are, due to their physiology, are able to explode in populations even when the water is relatively cold. Okay, so they come out really early, but the reason they start dying off here has to do with that silica. Silica is a silicone atom then with like 4 oxygens added to it. So, and they need this chemical, this molecule, to grow their shells, basically their cell walls. And silica runs out, and it's a really slow resource to rebound. It doesn't come back very quickly. So, the reason diatoms then come out early is because their physiology. And because then all the diatoms go away, there's kind of some empty space in the lake, and the green algae are growing at that time, and it finally gets warm enough for them. So the appearance of their coming out has to do with, again, that physiology, but then also basically there's lack of competition now because all the diatoms have already died off. So the green algae come out, and but the reason why they start to get reduced in late summer has to do with blue-greens. Blue-greens are really good at competition, but they need really warm temperatures. So the lake is pretty warm. Again, it has to do with the physiological constraints in the beginning, and they compete really well, and their numbers really increase. But then it gets too cold for them, and they start to go away. So, and then at that time, then the amount of silica, if you kind of were to make another graph of like silica abundance, it would, and same through time here, it would be really high and then go up, and then it would kind of slowly increase and then go down again. So this portion would be right here, and this bloom of diatoms here would be right in here. So when we look at then the whole phenology of this whole system, we see that there's really a lot going on here, from physiology, competition, resource availability, all sorts of different things going on that create the average phenology that we see in a phytoplankton community. So I kind of want to then move into this idea of what is a priority effect. Now, classically, we think of a priority effect as this inhibitory interaction between coexisting species. So maybe let's think about two species being equal competitors. Okay, we have species A and species B. And they have the ability to coexist at some time, or they're maybe I shouldn't say coexist, but maybe I should use the word maybe exist in the same habitat. Okay. And what's interesting is that if we have two equal competitors, sometimes it just matters of who gets there first. Okay, so if there's plenty of scenarios where we have a pairing of species that if A arrives first and gets a established population, A will essentially win and competitively exclude species B. But if species B arrives first, B competitively excludes species A. There's plenty of examples where this happens. And I just did a quick Web of Science search looking for ecological priority effects, and this was looking at ones where looking at an invasive grass species, and it kind of basically the ability of an invasive grass to get in depended on the native plant species, right? So the native plant species were inhibiting the growth of this invasive species. Was a really interesting paper I looked at that looked at priority effects and parasites, and what it was looking at human parasites, and they were looking at like numbers of parasites in the body, and what they found was that if one parasite were to be able to get into the host, they oftentimes inhibited other species from being able to effectively infect the host. And they wouldn't, the later arriving infections wouldn't be able to essentially gain a foothold. And so like all of that energy was being used then by the first parasite that got there. And then, there's all sorts of examples with tribolium beetles. These are wheat beetles, where they were looking at when food was limited, they had two species of these wheat beetles in their little jars, and if you put them in there at the same time, you can have a coexisting of these two species. But if you let the first species A go, species B won't be able. If you let species A go for a couple weeks, species B won't be able to make an established population, and vice versa. So there's lots of examples where we see priority effects. The timing of colonization matters for whether we can get a population going. And I've actually seen this in some of my own work. So this is an experiment I did looking at tiny little mesocosms. So this is, across here is about 40 inches. It was just your little kiddie swimming pool. And I was looking at playa wetlands. So what I did was I filled these kiddie pools with playa wetland sediment. And then I was trying to do an experiment because one of the things that I noticed with playa wetlands is they have very big differences in the amount of plants. And I wanted to, not too many things actually eat like aquatic plants or plants that are growing in there. They're more concentrated on the phytoplankton and the periphyton. So I wanted to see if adding essentially plastic plants would make a difference. So you see here is this is the just normal no plants, and this is these little plastic plants here. Now it's interesting that this no plants is looking very different than this bunch of plastic plants in here. But what I found is essentially it doesn't look like it here, but the plants didn't matter, Okay, I saw basically two states of a turbid water system. So we got, all this water really cloudy, and a clear water state. And it had to do with the number of triops, or tadpole shrimp. Tadpole shrimp are these, they kind of look like a tiny horseshoe crab. But what they do is they're really fast hatching, and they develop really quickly. So you can grow these in a little jar, and within you put the eggs in the water, and within literally 3 days you can have things about the size of a pea, and then after 6 or 7 days they'll be about maybe as big as your thumbnail and your first knuckle. So they're extremely fast developing compared to the rest of the community. And what they do is they dig around in the sediments. Okay, and what that does then is it puts a lot of dirt up into the water, and that's what happened in this situation. And what I saw was so the plants didn't matter in this mesocosm experiment, but what really mattered was the number of triops. Okay, the number of those tadpole shrimp. So if I had very few tadpole shrimp, this is what I got. Okay, so this had few triops in it. Okay, and if you can see, the water is extremely clear here. The water level is about the same in these things, but you could totally see the bottom of it. And there is just basically, maybe one, maybe two triops in this little container here. So yeah, they're like putting up mud and stuff into the water, but it would basically settle out. Whereas here, I maybe had 10 triops. Okay. And what happened was then the water got really turbid. Basically, because there's all those tadpole shrimp digging around in the sediments, it put those sediments up into the water, and then created a turbid environment. Well, so what that meant then is that periphyton would be dominant in the clear water scenario, which translated then to different a herbivore community that was mainly scrapers. Okay, so basically snails and little mayflies that could scrape the periphyton off of these little rocks that I put in here, and just on the top of the sediment. Whereas in the turbid water scenario, basically there wasn't any periphyton. It was a phytoplankton-dominated system, and there was more filter feeders found in this scenario. So when I see these mesocosms, what I was really, my experiment in some ways failed because it didn't actually get to test what I wanted, but I found this really cool interaction where the number of triops really determined what the whole community was like. The more tadpole shrimp that we had, the more turbid the water is, and the more phytoplankton driven the whole entire food web was. So it made a huge difference in the whole community. All right, with that, we'll leave that at that for now, and we'll come back thinking about assembly rules.